Welcome, everyone, to this webinar on CAR T-cell therapy organized by Myeloma Patients Europe. I'm Solène Clavreul, and I will be moderating this session. I will briefly introduce MPE and our speakers before giving them the floor. Myeloma Patients Europe is an umbrella organization of myeloma and AL amyloidosis patients groups across Europe. And we currently have 48 members based in 31 countries in Europe. And our mission is to provide education, information, and support to member groups and to advocate at European, national, and local levels for the best possible research and equal access to treatment and care. As you know or may know, CAR T-cell therapy is a new way of treating multiple myeloma, and this new therapy has raised a lot of hope and lots of questions within our community. So in particular, patients have expressed the need to talk to other patients who have already experienced this kind of treatment. And we have organized this webinar so you can all ask your question on CAR T-cell therapy. I'm very pleased to welcome our three speakers of the day, Dr. Michel Delforge, Professor of Medicine and Clinical Head of Haematology at the University Hospital Leuven in Belgium. Hello, Michel. And he will present how CAR T-cell therapy works, the different steps of the treatments, the first results obtained with this therapy, and we'll talk about side effects and impact of this therapy on patient quality of life. We also have with us William Kenneth Casler and Tamra Fine, who have both received CAR T-cell therapy in France and the Netherlands. They will share with you their experience and answer your questions. Thank you all for being there. Before we start, I would like to know what brought you there through this little poll. So this is a question for the audience to better understand what you're looking for joining this webinar. We have several questions. Please answer them and we'll share the results with the audience afterwards. While you answer the poll, I just would like to give a short reminder to everyone on how to use the Zoom webinar app. So attendees are not seen or heard in the webinar, so you should be able to see and hear the presenters, but you will not be able to see and hear other attendees, and they will not be able to see or hear you. If you cannot hear the presenters, make sure that your speakers are not muted and that the volume is set high. And if you're experiencing any technical issue, please let us know in the chat, and my colleagues will try to help you. You can watch the webinar in gallery view, and the gallery view setting enables you to see all the presenters in a tiled format on your screen, and the view setting can be changed in the upper right corner of your screen. You can toggle back and forth between gallery view and speaker view. You can use the Q&A question and answer feature, which is found on the toolbar at the bottom of the window. And please use this feature to ask a question to the panel. You can click on the like button to upvote a question and let us know what questions are of high interest to you. Don't wait until the end to ask your questions, because we will try to group them by topic while the presentation is ongoing. And if many questions remain unanswered at the end of the webinar, we will collect them and work on an educational resource to be shared with the community afterwards. You can also use the chat feature to chat with other participants, share your experiences or comments on the current discussions. And if you're, again, experiencing any technical issue, please let us know about it here, and one of my colleagues will assist you. You can consider audio only if you're having any troubles with your audio or video, a poor video could lead to a signal being cut off, so consider attending in audio mode only and bypassing the video if you're experiencing that kind of trouble. If you don't see the toolbar at the bottom of the Zoom window, please move your mouse slightly and the bar will appear, and the bar disappears after a few seconds when you are in full screen mode. The webinar is being recorded and will be available on our website at www.mpeurope.org and social media channels. We'll put the address on the chat. We will now share the poll results and see that apparently most of you want to know about everything. So we will try to meet these expectations. There's high interest into how CAR-T works and if it's efficient and safe, but also about the feedback from our patients there that are members of the panel. So thanks a lot for sharing your answers with us, and we will start with Michel's presentation. Michel, the floor is yours. Thanks so much for being there.
Thank you very much, Solène. Thank you very much, Myeloma Patients Europe, for this invitation, for a lecture that I consider as very important. And also I look forward to listening to the testimonies of Tamara and William. And of course, I look forward to the discussion. So I will now share my screen, and that's, as I mentioned, a little bit always a challenge because, yeah, I hope you can see it now. If not, Solène, you let me know because
it's working well.
Okay, excellent. Because we use more and more Teams and less Zoom. So, but I'm happy it works. So what I will do in the following 15 to 20 minutes is just give you the perspective from a hematologist treating myeloma patients, how, let's say, we look at CAR-T treatment. And for the time being, it is a treatment for patients with what we call relapsed and refractory myeloma. So patients who have received many other treatment lines, frequently who have exhausted all the available treatment options. And of course, for a patient to hear that your disease has relapsed is very bad news. But I can tell you, after so many years, and I had to do it even twice this afternoon, it's always difficult to tell a patient that there is a relapse of the myeloma, even after initial excellent response. But on the other hand, it's so important when you can give patients perspectives to be able to control the disease once more. Now, CAR-T cells, they, let's say, refer to our immune system. And the strongest, and I apologize for the word, but the strongest weapon our body has against invaders, whether they're viruses, bacteria, or cancer cells, is our immune system. So why does the immune system does not recognize cancer cells? Well, there's an awful lot of research being done, but it can be intrinsically related to the type of the tumor. And in terms of immunology, sometimes they talk about hot tumors and cold tumors, meaning that hot tumors are tumor cells that are easily recognized by our immune system, and other tumors are not recognized by the immune system. An example of a hot tumor is, for instance, melanoma. It's not myeloma, but melanoma, skin cancer, where sometimes the immune system gets control over the disease, and immunotherapy or therapies that activate the immune system are very well working. So it's already been studied for decades, like how can we use the immune system to fight cancer? And in that regard, vaccination is, let's say, a possibility, because if we want to prevent infections, we get a vaccine. So why not giving a vaccine against the cancer? And this has also been explored in myeloma, but unfortunately, it didn't really bring the results one was hoping for. So you make a kind of a mixture of myeloma cells, of plasma cells, and you inject some of the proteins like you do with a vaccine, and you repeat it several times in the hope that you will provoke an immune response. But the results are, despite a few decades of research, pretty disappointing. So alternatively, we could try, let's say, to manipulate immune cells in another way. And so this person, Dr. Donald Thomas, he received a couple of decades ago the Nobel Prize for Medicine because he was the godfather of the allogeneic stem cell transplantation. So in other words, you bring immune cells from a healthy person into the body from a patient, hoping that the immune cells from the donor will attack the cancer cells. And this is what we still do for diseases like acute leukemia, and which is also, but less frequently than in the past, being done for multiple myeloma. And also in my earlier years, we did several allogeneic stem cell transplantation, donor stem cell transplantation, trying to cure myeloma. And sometimes you see that indeed these immune cells from the donor, they can get control over the disease, but this is more occasionally. On the other hand, this is a type of therapy that can be associated with severe complications, where the immune cells from the donor start also to invade healthy tissues and attack healthy tissues like skin, liver, or the gastrointestinal tract. So looking in more detail at the immune system, we are very much interested in T cells. T cells are specific immune cells. And what I try to show you here is how T cells function. And T cells, they have a receptor, and you can compare it with a key, with a unique key that fits into a unique lock. And that unique lock is what we call an antigen. For instance, if you get a viral infection, you would get hepatitis, then the liver cells get invaded by the hepatitis virus. And as a consequence, these liver cells will express viral antigens. So let's say particular locks. And these will be recognized by unique T cells. So the key fits into the lock. And as a consequence, the cell will die. This is an extremely efficacious system in our body. And it is because of this type of system that our body has unique defense mechanisms against invaders like viruses, like bacteria. But it's a dream already from many investigators, from many clinicians, to use these T cells against cancer cells, and not the T cells from a donor, like we do in allogeneic transplantation, but to use the T cells from the patient themselves. Because if you have multiple myeloma, that's a disorder of B cells. There is nothing wrong, basically, with your T cells. But a condition is that you have a unique lock on the plasma cells, on the cancer cells, in other words, that allows the T cells for recognition. In other words, that the T cells recognize this unique antigen on a cancer cell. And so the first task is to find the right target. Because if you would have a target that's on a cancer cell, but also on too many healthy cells, like, for instance, on liver cells or kidney cells, you would bring the patient into big problems, because the T cells would attack and will attack all the cells that have this antigen or have this lock. So the good news is that in some diseases, particularly in hematological diseases, you can find quite unique antigens or targets for the T cells. And one of these targets in myeloma is called BCMA, or B cell maturation antigen. It's a target, it's an antigen that's quite uniquely expressed on a particular group of B cells, including plasma cells. And there's like no important or expression on other tissues. So in other words, that's a pretty unique target to direct the T cells, or for redirection of the T cells. But then the point is now how to do this. Well, biotechnology has evolved so impressively, and it's not a piece of cake, but over the years, this technology is well known and well controlled. So a little piece of DNA that codes for this BCMA is introduced into the T cell. That's what is called bioengineering. So the T cell gets a little piece of DNA inserted, and as a consequence, these T cells will make the key to detect the BCMA lock. So that's what you see here at the right side of my slide, and I'll just take a pointer. Here you see the normal T cell, and here you see this kind of, you could call it an antenna, I would call it a key, that is, let's say, made by this piece of DNA. So the cell will make this receptor. We call it an chimeric antigen receptor because it's a kind of a genetic construct. And as a consequence, this T cell will now at the surface express these CAR receptors, or in other words, the keys to detect the BCMA lock. So this is what we call a genetically modified CAR T cell. Now, these are pretty complicated slides, but what I want to show you here is just that by using this type of therapy, we really see unprecedented results on the disease, unprecedented efficacy. Because normally in this type of setting, if you would use another type of therapy, you would see that unfortunately, 1 out of 3 patients will respond and 2 out of 3 patients will not respond. And the duration of the response will be short. With CAR T cells, it's a different story. The majority of patients will respond, and a significant percentage of patients will have an extremely deep response. And this translates in a long response duration. And currently, we are faced with several anti-BCMA CAR T cells from different manufacturers. Some are made by non-commercial entities, by universities or university hospitals. And they try to improve the efficacy of the CAR T cells. And for instance, here you see with this particular CAR T cell product that nearly all patients who have, let's say, more or less exhausted all the other treatment options, nearly all of those patients, their disease will respond to the anti-BCMA CAR T. And the majority of these responses will be extremely deep. And in a significant number of patients, we will not find any trace of the disease, even if we use the most sensitive techniques to try to detect residual myeloma. This is very promising. Does this mean that this will be a cure? Well, of course, that's what we hope for, but unfortunately, even with extremely deep responses, many patients will ultimately relapse, but some patients will not relapse. And this, just as an illustration, this is one of the first patients that I treated with anti-BCMA CAR T. And I really had to sit down when I saw the results because it was so astonishing. And it's really black and white. On the left side, you see the PET CT scan of the patient at the time of progression. And the myeloma was really everywhere, nearly in every single bone. All the black is myeloma. And this was on the 23rd of October. He was infused with CAR T cells on 5 November, more than 4 years ago. And on the 12th of December, we had this control PET CT where nearly all of the lesions were gone. And this just shows the potency of this type of treatment. But there are also drawbacks. I mean, it's a pretty complex methodology, and several steps are already being taken to, let's say, to make it more easy. It's like, for instance, with cell phones, the first generation of cell phones is hardly to recognize when we look at the current generation of cell phones, but still this took us 1 to 2 decades just to have this type of revolution. And with CAR T cells, things are being sped up, but nevertheless, this is a complex procedure that can take up to 4 to 8 weeks between the apheresis, so the prelevation of the lymphocytes from the blood, and the infusion of the CAR T product in the patient. And during that time period, there can be progression of the disease. So we need to make sure that we are able to control the myeloma when the CAR T cells are manufactured. And before we infuse the CAR T cells, we also have to give a low dose of chemotherapy. And because the T cells are a tiny little bit modified, they could be recognized by the other T cells as invaders, intruders, and our other T cells could destroy the CAR T cells. So that's not what we hope for. So therefore, there is a small chemotherapy given the week before just to suppress the immune system to avoid that the other T cells would destroy the CAR T cells before they can exert their function. CAR T cell therapy should be done in a specialized center that's qualified for this type of treatment, where there is a whole team around the patient to make sure that this type of treatment can be given in the most optimal way. Because there can be side effects. And just think about a situation where you had, for instance, flu. When you have flu, you can have high fever, muscle pains, you can have headaches, you can feel nauseous, you can have to stay in bed for a few days because you really feel ill. And why is that? That's not because of the virus itself. That's because of your immune system, because your immune system is fighting the virus, because your T cells, amongst other immune cells, are expanding or growing and are reacting. So if we give CAR T cells to patients, we give it with a particular aim, that they attack the myeloma cells and that they grow, that they just expand to attack more myeloma cells. And this is some consequences. When T cells grow and are active, they make chemical molecules that we call cytokines. And these cytokines, they can cause symptoms. And one of the frequent complications of CAR T cells is what is called cytokine release syndrome, or CRS. And this CRS occurs in nearly all patients who receive CAR T cells. And it can occur on the day of the infusion, but sometimes it can also only start after 7 days of the infusion. So patients need to be closely monitored. In other words, the majority of centers, they stay hospitalized for 2, sometimes 3 weeks, just to make sure that the treatment can be given in a safe way and that complications like CRS can be diagnosed and appropriately managed. Because CRS is a kind of a systemic disorder, just like when you have a severe inflammatory disease like a severe infection. And fever is a typical symptom, but it can also be associated with a drop in blood pressure, with feeling short-breathed, with other, let's say, symptoms. So we as physicians, together with our nurses, we need to be very vigilant. We need to monitor the patients very closely. And if the CRS is severe, we need to intervene. Another complication that can occur in the first days after the CAR T infusion is what we call neurotoxicity. What does this mean? Well, you can have some of these T cells that make it into the brain, or these small chemical molecules that we call cytokines that are produced by the CAR T cells, they can also go into the brain. And this can cause typical symptoms, like patients that start to be confused, patients that start to have difficulties writing, they are losing their orientation, or very occasionally they can develop seizures. The good news is that with the anti-BCMA CAR T cells, this type of neurotoxicity is not so common. When we compare it, for instance, with the CAR T cells that are used in other malignancies like non-Hodgkin's lymphoma, where you see more of this neurotoxicity with the currently available CAR T cells. But it could happen that a patient who receives BCMA CAR T cells gets confused or develops other neurological symptoms. And then we as physicians also need to intervene. A third complication that is very common when patients receive CAR T cells is that the bone marrow is suppressed. Part of it is related to this chemotherapy that we give the week or a few days before the CAR T cell infusion, but part of it is also caused by the inflammation that the CAR T cells cause, and that can have a suppressive effect on the other bone marrow cells. So patients have a higher vulnerability for infections. And even when the acute phase is over and when patients go back home, there can be an increased risk for infections even for months. So patients need to, let's say, take appropriate measures, precautions for prevention, and also have a low threshold for contacting their healthcare providers. But finally, and I think this is, let's say, also a very big milestone of CAR T treatment, that it's just one infusion. And okay, then you go through this period of side effects, but then the quality of life is so much better. And many patients will tell us that their quality of life is so much better than they ever had with all the different treatments. And this is what you see here with these green bars. So this is before and after for different, let's say, symptoms like fatigue or pain. And you see here that the physical functioning also and the global health status that patients score themselves, they is so much better after the CAR T treatment. And this is after a year after the CAR T treatment than at the time of the CAR T infusion. So in conclusion, I think it's beyond any doubt that CAR T cells, they are just a masterpiece of innovation in biotechnology and medicine in the benefit of patients. Currently, we have two BCMA targeting CAR T cells, ID-cell and cilta-cel. They are both approved by the FDA and the European Medicine Agency for treatment of relapsed and refractory multiple myeloma. They show unprecedented anti-myeloma responses, but nevertheless, patients might relapse despite impressive initial response. But this is just the beginning of a new journey, and we just have to wait long enough. And I have no doubt that there will be many patients who will not relapse after successful CAR T treatment. Currently, we use particularly the autologous CAR T cells. So they are derived from the T cells from the patient themselves, but there are also studies ongoing with donor T cells. So in an allogeneic setting, but again, here you might have the problem of rejection. So this requires additional genetical modifications of the CAR T cells. But as you understand, this is a very complex and also time-consuming manufacturing process, although the time between the prelevation of the T cells and the infusion will become much shorter in the following years. Infusion of CAR T cells should be done in a specialized center, and it is mostly associated with side effects that are pretty unique, like cytokine release syndrome. And there can be also prolonged suppression of the bone marrow and increased risk for infection. And there are currently ongoing trials that explore the role of CAR T cells in earlier phases of the disease. And last week, there was a press release from a major publication that has compared one infusion of CAR T cells with long-term treatment in patients with relapsed myeloma. And currently, there are studies ongoing where CAR T cells are given as part of the initial treatment in newly diagnosed patients, and we are eagerly awaiting also those results. So thank you for your attention. I will now stop sharing my screen.
Thank you so much, Michel, for this very comprehensive presentation of CAR T-cell therapy. It was also very clear. Thanks. I'm sure our attendees have many questions for you, so please, if you are in the audience, share your questions using the Q&A feature so we can address them during the Q&A session. So now we'll hear the patient experience, starting with Tamra Fine. Tammy, the floor is yours. Thanks for joining us today.
Thank you. Happy to be here. I received my CAR Ts. I received the Abecma or ID-cell CAR T in a clinical trial at Erasmus University Medical Center here in Rotterdam. Lucky for me, it's where I've been treated for my myeloma, and it's very close to where I live. I received it after third relapse. So I have been, next month will be 10 years since I was first diagnosed. And I would typically get about a very good response and a very fast response to treatment, but then relapse after 2 years. So when I relapsed after daratumumab with pomalidomide and dexamethasone, the CAR-T trial was open and I was able to get in. And thankfully, I was randomized into the arm that received the CAR-Ts. I was really, as soon as I heard about CAR-T, I knew I wanted this because it truly is one and done. You go in my case, to the hospital, and in a clinical trial, they kept me in for 3 weeks to observe afterward. Much, much easier than stem cell transplant. For any of you who've had stem cell transplant, I can tell you, there's just no comparison. It's a much milder lymphodepletion that precedes it. And the recovery period was much, much faster. I happily had a very good response to this. I am MRD negative. It's been almost 2 years since I received my CAR-Ts. I've had no treatment in all that time, no further treatment, but a lot of follow-ups. So they monitor me every month, and I've had many, many bone marrow biopsies, including one today, which I await those results, but no signs of the myeloma anywhere, blood, scans, or bone marrow biopsies. So I am thrilled. My life has returned to what it was before diagnosis. And I feel like I did 10 years ago before I was diagnosed. I did have very mild grade 1 cytokine release syndrome following the CAR-T on day 3 after I had the infusion. It was like having a flu. I was feverish and achy and sweating and then chills. And when I didn't respond to paracetamol after 24 hours, the doctors gave me 1 dose of tocilizumab, which within an hour brought my temperature back down to normal. And I had no further problem. That was the end of the cytokine release syndrome. I had none of the neurotoxicity. I really never lost my energy. Even in the hospital, I had my laptop, I was working, I was using the exercise bike and had a PT to keep the muscle strength, because being in for 3 weeks is a long time. I joked with my nurses that it's a good thing I couldn't open my windows, because if I could, I would have tied my bed sheets together and rappelled down from the 9th floor to escape into the beautiful spring weather. So I have to say, for me, CAR-T has been a life changer. My work involves a lot of travel, and I've been able to resume that. I've been on African safaris. I hike in the Swiss Alps. I go skiing. I do absolutely everything I did before this little detour in my life called multiple myeloma. And I'm just really grateful for this. If I relapse and CAR-T was offered again, I would absolutely do it again. Thank you.
Thank you, Tamara. That was very clear. I'm glad that it was such a positive experience for you. And please, if you're in the audience, share your questions through the Q&A feature. And let's now hear the experience of William Kenneth Casler. Ken, the floor is yours. Thank you.
Thank you so much, Solène. Thank you. And thanks to Dr. Delforge for his excellent educational presentation. I thoroughly enjoyed it. And I'm very pleased to share my experience. I was very interested in listening to Tamra, because everybody has a different experience. In late December 2021, I woke up in the intensive care unit of Saint Louis Hospital in Paris, where I live. I was stricken by acute pneumonia and septicemia. And this was just the latest of my myeloma struggles since diagnosis in 2009. By late 2021, matters were becoming progressively more difficult for me, and I was penta-exposed, triple refractory with 4 lines of treatment. And that, of course, as we've heard, is the current profile for CAR-T cell therapy. So when I saw my hematologist again in January 2022, she asked how I felt about CAR-T therapy. And I immediately replied, yes, please, I'm in. And I was all the more excited that the French Health Technology Agency, which gives its approval to reimburse CAR-T, had decided that without a comparator, the cost-benefit analysis of the treatment could not be demonstrated. That's a disappointing bit of news. But despite the decision, there was still some 12 slots a month left in France for myeloma patients, very advanced myeloma patients. And I was fortunate to be chosen, I'm fortunate to be among the chosen few. So in early February, I entered the protocol only to encounter the first hurdle. My right ventricle had been damaged by years of intensive myeloma treatment. That's essentially chemotherapy and all the different drugs. The hospital's cardiologist was not keen for me to risk CAR-T therapy. And after lengthy negotiations between the cardiologist and my hematologist, I was put on an ACE inhibitor and a beta blocker. And if you've ever experienced that, you know it does nothing to enhance your quality of life. And to say that I was a party to the discussion is a bit of a euphemism. The cardiologist made it clear, take the medicine or forget the CAR-T. And who was I, a mere patient, to have any say in the matter? And need I say, I did not appreciate the attitude. And by the way, I never saw the cardiologist again. He simply disappeared from the radar screen. Quite amazing. Although my myeloma was progressing again, slowly, I did not require bridging therapy. So in early May, the lymphodepleting chemotherapy was administered. And I found it I got ahead of myself. The whole harvesting of lymphocytes took place in mid-March. And I found that much more easy to tolerate than the stem cell harvesting that I had experienced in 2010. In 2010, I spent 5 days, 5 hours a day on the machine to harvest the stem cells. And here for the lymphocytes, it was just one morning. So my lymphocytes were sent to the USA, where the factory is, for modification and growth. And as the doctor explained, it usually takes 4 to 6 weeks. And in my case, it took about 8 weeks, took a bit over 8 weeks. And I think anybody who's been heavily treated for myeloma will know that medical delays really are among a patient's worst enemies, because they create a space for our minds to wander and to create scenarios of dread. And so, they can be challenging. And fortunately, for many years, I've been practicing meditation and yoga, and I've learned to manage such uncertainties with equanimity. So being able to balance my mind and regulate my emotions. Finally, in May, on the 17th, I was admitted to the hospital for treatment. Again, the usual battery of tests, cardiologist, neurologist, and then the lymphodepleting chemotherapy was administered over 3 days. Then I was given 2 days of rest before infusion of the modified CAR-Ts on day 6. My prior experience of 2 stem cell transplants in 2010 and 2018 prepared me well for CAR-T treatment, and I knew what to expect. But what I didn't expect was the roller coaster ride of side effects. Maybe it was my age, I'm 75, or just a faulty memory of 2 earlier bone marrow transplants, but I found the 5 days after infusion a little challenging, so quite different from Tamra. I experienced grade 1 CRS, high fever, 41.5 degrees centigrade, grade 2 thrombocytopenia. My platelets dropped to 12,000, requiring 3 transfusions. There was also severe anemia. Hemoglobin dropped to 7.6. I also needed 3 blood transfusions for that. I was quite keen to record my CAR T experience, so I took a diary and kept notes. There's not a single entry for 3 days after the reinfusion. I had ICANS. One day I was sure I'd received an email from my hematologist authorizing me to step out of my room for some exercise. A kind nurse gently reminded me that I was not allowed outside my room just yet and took me back to the sterile room. And I like the image that Tamra used of tying the bed sheets together and climbing out the window. I would have done the same. So after 16 days in the hospital, I was released and happy to go home for some decent food. I had lost 7 kilos. I was also keen for a little exercise. For many years, I've made a point of putting in 2 hours of exercise a day, some combination of yoga, tennis, bicycling, and walking. How great was my disappointment when I discovered that I had 0 energy. Yet, a week after my release, I recorded 2 video interviews for patient organizations about CAR T treatment. I managed a trip to Bordeaux to see my grandchildren. I even went to an MPE masterclass in Amsterdam, all within a month of the CAR T therapy. My self-confidence and exuberance were really over the top. I was, of course, dead tired after this somewhat reckless running around. But worse, I soon stopped eating and began suffering from severe diarrhea 10, 12, 15 times a day. And over a 3-week period, I lost another 8 kilograms. And I was readmitted to the hospital with COVID. My running around had been an extremely bad idea and exposed me unnecessarily to a heightened risk of infection during recovery, just as Dr. Delforge warned. I'd been seeing my medical team on a weekly basis. They quickly identified COVID and I was administered Paxlovid, but that only hid the symptoms. And finally, after 10 days in the hospital, in late July, the COVID was brought under control with convalescent plasma. At one point in early August, I was taking 11 drugs. There were also growth factor injections and continuous blood transfusions. I remained weak until September, at which time I weighed 55 kilo for 1 meter 82. There is a happy end to this CAR T story. In November, my hematologist informed me that I'd achieved very good partial response. Yes, some myeloma still lurks in my system, but in smaller amounts, and the M spike is below 2, and the light chain's unquantifiable. But frankly, that's a bit irrelevant. What matters most, in my opinion, is my sense of well-being today. And on that score, I can honestly say, like Tamra, I have never felt better. I have no immunotherapy treatment for myeloma whatsoever, though I do continue to take antibiotics and antivirals and a medicine for my heart. So the one and done slogan applies to some patients and to myeloma treatment, but not to all medication. I'm negotiating to be drug-free by next summer. I hope I succeed. With my return to normality, I have resumed my full range of activities. Next summer, I plan to be back on my touring bike. for some long distance cycling, hopefully on a pilgrimage path, which is called the Via Francigena, which I've done halfway, and I hope to ride from Aosta in northern Italy to Rome. And then the following winter, I hope to be back in Southeast Asia again on my bicycle. Projects of any kind are an inspiration. They help me to project forward into meaningful activities that involve engagement in life. They also enable me to sustain my curiosity and experience sort of awe and wonder. I like to go and explore new places, different places, be open to these different experiences. So on that score, my experience of multiple myeloma has been transformative, and it's enabled me to appreciate the marvel of life and the goodness of people to a fuller extent. And I thank my family and my friends for accompanying me on this journey. So with my return to better health, I'm able to once again embrace all of life's opportunities. And that, as far as I'm concerned, is certainly worth striving for. Thank you for listening to my story. I hope I wasn't too long, Solène.
You know the answer to that. Thank you, Ken, for sharing your experience, for keeping track of everything that you've been through since you started that treatment. And I will invite Tamara to turn her camera on so we can see all the panelists and answer the question together. We are now reaching the end of these 3 presentations. Many thanks to our speakers. We will now move to the question and answer session. And please, if you are in the audience, add your questions to the Q&A window, and we will try to answer all of them. So I will start with more clinical questions for Michel. We have received a question, maybe to start with, to clarify how does CAR T-cell therapy differ from monoclonal antibody therapy? I think we have many more clinical questions coming, but maybe just clarification around the definitions and what we're talking about would be appreciated.
Yes, thank you, Solène, and I will do that. But first of all, also let me congratulate Tamra and Ken for their, let's say, astonishing how to say it? Their testimony. Sorry, and I heard Ken also a couple of days ago in Rotterdam, in the Netherlands, at the EHA EBMT CAR-T meeting. But I think yes, so much thanks to both of you for your testimonies and also for your positive messages of inspiration also for other patients. So now coming back to the question, how CAR-Ts are different from a drug like daratumumab. Well, we call a drug like daratumumab a monoclonal antibody, and it's a form of what we call passive immunotherapy. So it's a drug that you deliver, and daratumumab is an antibody that will bind to the plasma cell. But then it's, let's say, not for granted what then will be the next step, because the drug itself, by binding to the cell, might kill the cell, or the drug can, let's say, mark the cell for other immune cells. So it's a kind of a marker or a kind of a magnet that binds to the cell, and that kind of recruits or activates some immune cells. And so, but the drug itself, it's just there, and it can have some activity on its own, but part of the activity is depending on the immune system. A CAR T cell is what we call active immunotherapy. So it's a cell, it's living tissue, and it's a genetically modified cell, but it's a living tissue. And so once you release it into the bloodstream, it will start doing its own things.
Thank you, Michel. My next question is also for you. it's about the timing when T cells are harvested. Wouldn't it be better to harvest them when the patient is in remission rather than waiting until relapse?
Yeah, that's a very good question. And there is a lot of research currently ongoing about this topic. What you want to avoid is that you do an attempt of harvesting these lymphocytes, these T cells, at a moment when their number is very low. For instance, related to a previous chemotherapy treatment that kills blood cells, including lymphocytes. And there have been situations where the harvest was insufficient to generate enough CAR T cells. Secondly, there is not only an issue about the number of the lymphocytes, but also about, let's say, their function, their activity. And in that regard, of course, when you have, let's say, higher number of T cells that have not yet been exposed to any kind of chemotherapy, that's of course what you want to prefer. But on the other hand, we see that just by acknowledging the current data and just by listening to the testimonies of Tamra and Ken, that even if there has been a lot of exposure to these cells by previous treatments, that still it can be highly effective. And secondly, if you would start collecting lymphocytes, let's say in all patients, without any, let's say, certainty or any guarantee that later on CAR T cells will be needed, then you end up with also storing cells, which comes with storage capacity, with cost when it's not needed for a very high number of persons. But it could be that in, let's say, in a few years from now, when CAR T cells or CAR T cell treatment will have, let's say, a more stable place in the myeloma treatment, that we will collect these lymphocytes earlier in the treatment journey.
Thank you, Michel. I have another questions about when this treatment can be taken. You've talked about the context of a heavily pre-treated patients. When do you think this will come to patients at earlier lines of therapies or even at first line in the future?
Yeah, also another very good question. It is a stepwise approach, and this has been the case for all the other myeloma drugs. Also with, for instance, daratumumab started, let's say, in monotherapy at the end of the treatment journey, and now it has been already accepted for a couple of years as a frontline treatment, and this in less than a decade. But with CAR T cells, it's similar. First, you have to prove that it works, and that proof has been given without any doubt. And then the next step is to do studies in earlier lines of the treatment, but to do comparative studies, because that's, of course, what regulators require, but also what we as physicians want to know, if the treatment is better compared to the current standard of care. But in patients who have exhausted all the other options, there is no standard of care. But I can tell you, some of the regulators, they don't accept it, or they do not easily accept it, because it's not a comparative study. So this requires a lot of debate and arguing. So it's pretty complicated given the very high cost of CAR T treatment. But the good news is that this process is going very fast, because the studies in relapsed myeloma have been done, and we know already part of the data. Let's just remind you of the press release from last week, where we see superiority of CAR T treatment over standard treatments in relapsed myeloma. And the studies for newly diagnosed patients are ongoing, but still this will require still a couple of years before we have the final readout of these studies.
I see. Okay, thank you, Michel. We've received several questions regarding where we could get CAR T treatment, especially for patients who don't have access to CAR T in their country of origin, if it's not reimbursed there. So how can, first of all, maybe where is it accessible right now, if you have the answer, in broad Europe? And second, if it's not accessible, how can patients from those countries access this treatment somewhere else?
That's a very good but a very difficult question. And at this point in time, we need to say that the demand for CAR T treatment largely exceeds the availability for patients. And there are several reasons for that. First of all, this is very complex, as has been illustrated in the previous talks. So the manufacturing is very complex. And we see that currently the pharmaceutical companies that produce these CAR T cells also are struggling with some issues regarding production. Part of it is related to COVID, but we see that currently there is, let's say, less product than was initially hoped for. Part of it is also related to this kind of factors that they need, and there is a very scarce, there is a lack of these factors, these supplies for making the CAR Ts on the international market. A second important point here is that these treatments are very expensive. So regulators really scrutinize these files for submission, not because of, let's say, the efficacy, but because of the extremely high cost. And I can tell you, I can only speak for my own country. In Belgium, currently we don't have access to CAR T. We had outside of clinical studies, but the clinical studies in relapsed refractory myeloma, as we call it, have been done. So as Ken illustrated, in France, for instance, there is limited access for some patients also outside of clinical studies. But this is very different from one country to another. And I felt very sad over the past years when I got many emails from patients in high medical need asking how they could get access to CAR T treatment in my center, in my country. And most of the time I had to answer that there was no possibility, which really didn't give me a very good feeling. I think Ken has also some comments to make here.
Yes, but before I will just ask you one follow-up question about this that came up in the Q&A window. Regarding manufacturing, you've talked a little bit about it, but do we have centers here in Europe for the manufacturing of those CAR T cells? And what is it going to look like in the future?
Yeah, it's clear that this is definitely an important alternative compared to the commercial products. So there are several initiatives. Some of these initiatives are taken at the level of a local center. Some of these initiatives are taken at the level of a particular country or a region. And also at the level of Europe, there are, let's say, collaborative networks to manufacture CAR T cells in order to give more patients access to CAR T treatment, also at a lower cost. But again, also this is not easy, because to manufacture CAR T cells, you really need to have a very specialized lab environment, and the investment is many millions of euros. So not every center has this, let's say, opportunity. So, and secondly, it's important to provide a product that also fulfills the highest quality or criteria as commercial CAR T-cell products. And finally, it's also important that there is a payer. And this system, as we all know, is so different from one country to another, and in some countries, even from one region to another, or different from the health insurance that patients have.
Okay. Ken, do you want to comment on this?
Yes, please. Thank you, Solène. I just wanted to come back to the issue of patient need and cost of treatment. Referring to France, we have about 5,400 newly diagnosed myeloma cases every year, and the patient community is somewhere around 30,000, 32,000 myeloma patients. Last year, commercial CAR Ts were available for just about 100 people. The minimum need is at least 700, probably double that at present. So we have a real problem of access and availability. I was happy to meet the CEO World of BMS a couple of months ago on a one-to-one occasion, where he talked about the future of myeloma production. We're moving rapidly towards automated facilities, which should, with the help of artificial intelligence, increase the capacity and hopefully meet the need. But now there are all these issues of comparator that cannot be ignored, but they're ramping up the manufacturing processes.
Could I
As to cost, just very quickly, as I said, I've been treated now for 13 years, and I did a back of the envelope estimation of what my myeloma treatments have cost the French healthcare system over the past 13 years and my 5 treatments. And I've reached a figure of close to 1 million euros. Now, it's true that CAR T is expensive. I think it costs about 340,000 euros in France. But if I was on pomalidomide for 3 years, and that's basically 300,000 euros, I had 2 stem cell transplants, 180,000 per stem cell transplant. And then, of course, daratumumab, carfilzomib, dexamethasone for 2 years as well. And you add it all up, and that's almost 1 million. You move it forward, if we can get the approvals, you move it forward to the front line, at least for the high-risk patients, and you will make a huge cost saving. Somehow the HTAs, the Health Technology Assessment Agencies, are not getting that message. They're sort of camping on their position of, we don't have a comparator.
Yeah, and I, as you did, Ken, I did just a sort of back of the envelope calculation that they've now reached break even with me on the CAR T. What they would have been paying out for maintenance and treatment and then maintenance, it's a wash. So it's upfront investment, and then everything now is gravy. They're ahead of the game on what they're spending on me. The other thing I wanted to say, and maybe Dr. Delforge, you know something more about this, the fast CAR T, that turnaround time in 1 to 2 days is on the near horizon. Is it in China, or where is it that it's coming from?
Yeah, I mean, and it was also presented at the last American Society of Hematology meeting in December. So it's not only limited to China, but if we look at the global scale, we see that the highest number of clinical studies with CAR T cells, and not only in myeloma, but also in myeloma, is being done in China. And but overall, and I'm not an expert in the bioengineering, but the way I look at it as a myeloma physician is that technology is evolving so fast, and this is very positive. And this is not only about, let's say, making the CAR Ts more effective, because you have CAR Ts now that are directed towards two of these antigens or two logs. You have CAR Ts that or cells that lymphocytes that, let's say, the most effective type of T lymphocytes. And then, as you mentioned, Tamra, also these production that can be the vein to vein time, as we call it, that can be largely shortened in the benefit, all in the benefit of patients. But for me, and this can sound a little bit naive, what we definitely need to do as a society, together with regulators, is to find ways of, let's say, dealing with it the right way, because traditionally it's you do a study in relapsed refractory with one product and then you bring it stepwise forward. But now there is so much happening all at the same time. And if we do this, let's say, in the classical approach of one step after another, then it will cost us an awful lot of time, because we have this, let's say, most effective CAR T cells with the shortest production time. Although here I think that companies have the resources probably to implement this technology on the existing CAR T cells. But the positive news is that this field moves forward so rapidly. One of the challenges is that's to bring this type of innovation as soon as possible to patients, all on condition that we prove efficacy and safety, of course.
Time is money, we say.
Yeah.
But time is lives, because the patients can't wait. That's the bottom line, you know.
I'd like to come back a bit on availability. Michel, could you tell us where in Europe the treatments are available outside of clinical trials?
Yeah, I mean, I cannot also give here a straightforward answer, because over the years, as I mentioned, we had requests from many parts of the globe, even patients from Australia. I remember a patient from Australia who was desperately willing to come to Belgium to get CAR T treatment. Alternatively, I have referred some of my patients to Germany to try to get access there for CAR T. But this was not always so straightforward, because also the local CAR T programs have specific, let's say, requirements. So it's not something at this point in time that we can say, okay, I don't get it in my country, so I will move to another country and ask if I can get the CAR T treatment there outside of a clinical study. Or if you would have a health insurance that's willing to pay for it, then of course you could give it a try and see if you can have the treatment in a country where it is licensed and where okay, your health insurance coverage is willing to pay for it, including all the hospitalization costs. And I even tend to forget, but over a couple of years ago, we even considered sending patients to China. That was just before COVID, to have their CAR treatment there. But to make a long story short, I can tell you, and I don't want to sound negative here, but it's not so easy, because in the same time, the myeloma is progressing, the clock is ticking, and you see that in many centers, in many countries, there is already a very limited access. So this creates even more complexity to give access to patients from other countries. But I think you can always try it. And there I would probably believe that maybe yeah, the professional societies can give their advice.
I see. Ken, you want to comment on that?
Yeah, just a quick one. I recently was involved in a webinar in the United States where similar problems of access and availability exist. And it was interesting to hear the doctor say that he recommends that patients enroll for a CAR T in a clinical trial as well as the commercial product, and they take whichever one comes up first. Now, in our European countries, that may not be so obvious, as Dr. Delforge indicated, that a lot of the clinical trials are over. But some of these big trials, Karma and Cartitude in the United States, which are now comparing use of the treatment in earlier phases or earlier lines, there are quite a few clinical trials. And so there is that choice, at least in the United States. And I don't know if it exists in Europe, but I have my doubts.
But yeah, and thanks, Ken. So and an additional comment, and I think it's also important to let's say mention this. Even this webinar is focused on CAR T treatment, but there's nothing worse for a patient that if you know that there is a good treatment that could help you and you have no means of getting access to it. And I can tell you, I have been in a situation where patients were willing to sell their house just to be able to pay for the CAR T treatment. So, I mean, that's definitely not something we could, let's say, consider ethically. So for the time being, there is also the growing importance of a good alternative, which is called the bispecific antibodies. And that also target BCMA. And I had one slide that was about the side effects where there was this concept of a bispecific antibody, and where daratumumab that we just previously discussed is what you could call a naked antibody. So the antibody just moves towards the target. A bispecific antibody is an antibody that on one hand binds to the target, but it brings the T cell, it binds with another part, the T cell, and it brings the T cell next to the cancer cell, next to the myeloma cell, with a very high likelihood that the T cell will kill the myeloma cell. And we currently see that the results with these anti-BCMA bispecific antibodies, they're pretty comparable with the results obtained with a CAR T product like ID-cell. And so a couple of months ago, that's Janssen, which is manufacturer of one of the CAR T products of cilta-cel, and also manufactures one of these anti-BCMA bispecific antibodies, has launched an early access program in many countries in Europe to allow patients who would, let's say, need a BCMA CAR T cell to get access to these bispecific antibodies. And I think that's really also a major step forward. And we as physicians, we should not let patients down. We should always try to find a solution. And this really gives us a big window of opportunity to start treating patients with a BCMA targeting drug that works in a majority of patients, in the group of patients who have no access to CAR T. And in the following years, there will be, let's say, CAR T cell will be, let's say, the first option, but there can be reasons, and this has already been discussed, that the patient is not a candidate for CAR T because of, let's say, other diseases, because of the speed of progression of the disease. And plan B is then these anti-BCMA bispecific antibodies, where plan B is not so much, let's say, lower in efficacy compared to plan A. And that really gives us, let's say, the different treatment options to help the majority, the large majority of patients with relapsed myeloma. Awaiting, of course, these results of CAR T cells and also these bispecific antibodies earlier in the treatment. And I'm also fully aligned with the comment made by Ken and Tamara that in a couple of years ago, and that's also something that I try from my perspective bring to the attention of regulators, that we should try to look ahead. Regulators sometimes look just 1 or 2 years ahead, but I think I try to tell them, like, okay, this innovation is something that will help us to cure myeloma or to cure more and more patients with myeloma, but we should really, let's say, look at it in a more holistic way and not see it like, okay, this is one part or one treatment for a disease where we already pay an awful lot of money for all these expensive cancer drugs. But I can tell you, and I think you will agree, these are not easy discussions, but we really need, let's say, to hit the nail and in trying to let's say get this innovation sooner to more patients.
Definitely. Thank you, Michel. I'd like to move away a little bit from the access issues and talk a little bit about what will happen after CAR-T. We have one question around the length, the duration of the vulnerability for infection after the CAR-T treatment, and one after what happens after relapse from CAR-T.
Yeah, regarding the infections, now that's usually manageable, and I think that it can nicely illustrate it. Is that he experienced it a harsh way, and I mean, you could have attracted COVID also, let's say, closer to home, but still, it's something we should be very careful about. And the first months after the treatment, patients really need to be very careful. It's not that you need to stay isolated at home, but we really recommend patients to wear masks when they see other people, especially when it's inside, to regularly measure their fever, their temperature, and when they have fever, to have a very low threshold in contacting their nurse or physician. But so that's something that you also see with the bispecific antibodies. So they suppress part of the immune system, and so you need to take appropriate measures. There are patients who benefit from these regular infusions with immunoglobulins just to boost the immune system. And then secondly, regarding relapse, of course, that's, let's say, a very big disappointment. And I can tell you that one of my patients who really was as enthusiastic as Tamra and Ken, and then unfortunately she relapsed and she fell from a high mountain very, very, very low. And but still we try to find another type of treatment. It's not easy, but okay, there are some other drugs that are, let's say, accessible for patients who relapse after CAR T cells. You could even try to give them such a bispecific antibody, and there are a lot of data showing that the bispecific antibodies might work in a majority of patients who relapse after CAR T cell treatment. I think Tamara has also a comment.
Yes, I was just going to say that I read or heard on a webinar that they're finding that sometimes with the long pause that you have from a treatment you had previously and had become refractory to will work again after this time period of some years away from it. So that is, I've been told, another potential option after CAR T.
Yes, indeed, there are options, and it could be sometimes that when you have let's say another treatment in between, that you could reuse a drug from before. I think there what we physicians need to do is to be creative and see the patient in front of us and see, let's say, what is the type of relapse? Is it an aggressive relapse? Is it a slow relapse? How is the patient feeling? What were particular side effects of previously given drugs? How active were they? And what are potential alternatives, drugs that patients did not yet receive? And then just discuss this with the patient and then try to take the best decision possible.
Thank you both for your comments. Yeah, let's keep hope, and hopefully new treatments or previous treatments will will help after relapse. I have one last questions maybe before we close the session regarding efficacy. We have one around high-risk cytogenetics and one about the current results. So maybe can you tell us, Michel, who benefits the most from CAR T-cell therapy, and do we observe the same results among patients who are high risk or other kind of risk?
Yeah, also an excellent question. So I think every patient who has exhausted all the other treatment options for the time being, whether it's high risk or non-high risk, but also when patients have received many treatment lines, it's still called myeloma, but the biology of the disease is pretty different, or can be pretty different, from how the disease behaved when it was diagnosed. But what we do know, if that patients with what we call high-risk myeloma, and that's mostly driven by specific genetic abnormalities in the plasma cell, that those patients will exhaust the available treatment options much faster. And of course, they will be, let's say, earlier in the need for this type of treatment. What we do know from the currently available data that are published is that CAR T cells are also working for patients with high risk. But if we are honest, it's not exactly to the same extent as in patients without high risk features. So we really need to make more efforts to optimize the treatment for high risk patients. And for those patients, I'm a strong believer that we should introduce these innovative forms of immunotherapy much earlier in their treatment journey, and not waiting until you end up with a disease that is like a dragon with 7 heads, where you knock off one or you cut off one head when there's still 6 other left. So that is indeed a big challenge, yes.
So Len, may I come in on that?
Of course.
Very point. When I was first diagnosed with myeloma, October 2009, the cytogenetic analysis indicated a 1Q gain, 4 copies. Now, when we look at the cytogenetics, not only do I have the 1Q gain, but I have the T414 deletion, the T1114 deletion, and the T17 deletion. Myeloma changes its face all the time. And it's really, in French, you say course poursuite. I mean, you're just chasing after the thing and trying to target it, because it's a moving target. It's constantly evolving. And that, just if I may, I'd like to raise the question of how are we selected for CAR T therapy? There's no transparency, and as far as I can tell, how did the doctor decide that I, amongst the many hundreds of people in France, should get that therapy? Can you help us on that, Dr. Delforge?
Oh, I think, Mr. Casler, you should ask this question to your treating physician.
I didn't get an answer.
Yeah, but I can tell you, and in my experience, what we try to do is to take, let's say, the window of opportunity. And unfortunately, fortunately, I had patients that I could give access to CAR T treatment just the right time. But I also have to admit that there were patients who I could not give access because just for sometimes tiny little things, because in the study, they required a kidney function above a certain limit and the patient was just below. We tried, we negotiated, but we couldn't get access, or that the patient relapsed just a week after the trial had been closed. So I think sometimes it's also like with many things in life, you need a little bit of luck to be, let's say, not to have a relapsed myeloma, but when it relapses, that it relapses at the right time when there is access to this type of treatments.
Thank you, Michel. Well, we are reaching the end of our webinar. I'm so sorry we haven't been able to answer all questions, but I hope the answers you got were useful. We will collect all the unanswered questions and try to put them together and write some educational material with the answer to those questions and share them with you. Before closing the session, I would like you to answer one last poll to better understand how useful the webinar was for you. Michel, Tamra, and Kenneth, it was a very productive session. We all have learned a lot today. And on behalf of MP and all our attendees, many, many thanks for being there with us today, for taking the time to share your experience and your expertise, and of course, to answer all those questions. Do you each want to share a last word with our audience, a take-home message before we close the session?
I leave it up first to Ken and Tamara.
Tamara.
Okay, just I would say, never give up hope and keep on going and stay engaged, stay educated, educate yourself about this disease, about the treatments that are out there, about the treatments that are upcoming. There are a lot of good resources that you can find, groups like MPE and some others. Multiple Myeloma Research Foundation is the go-to place for me. Just stay with it, because there's so many things happening and so many good things coming up.
Thanks, Tamara. Ken?
I agree totally, Tamara. I'll just use slightly different vocabulary. Stay resilient. You're going to face adversity, but stay positive. Keep an open mind. Try to learn as much as you possibly can. Accept what's happening. You can't fight it, just accept it, live with it. Do not stop living. Enjoy now. And that's, I mean, the important thing is you're alive and you're happy now. So stay there.
Thanks. Michel?
Well, I think this is so inspiring, and thanks so much to Ken and Tamara, because I think it's our only patients who can, let's say, express in the most honest way, because they experience it. And but on the other hand, after let's say treating myeloma patients for nearly 20 years, it's still a disease that motivates me every day. And what motivates me is the patients who have the disease. And also from our perspective, we should not give up too early, and we should really try together with patients, with each individual patient, to try to find the best solution for their specific problem. And innovation is really moving very fast. And it's not by chance that myeloma is one of those cancers where we, let's say, see the benefit of innovation very clearly, and it will even get better in the near future.
Thanks. Well, that's very helpful. Thank you again to the three of you, and thanks as well to the entire MPE team for making that webinar possible. And I wish everyone a pleasant evening and a good Valentine's Day.
Thanks so much, Solen. Thanks to
Thank
MP.
you.
Thanks to
Bye,
everybody.
everyone.
Thank you, Solen. Goodbye.